Point mutations are the result of a substitution of one base for another, can have several effects on the structure of a peptide chain that is formed during translation. Here we have a DNA coding sequence. In order to be able to properly turn this DNA sequence into a peptide chain, we must first turn this DNA coding strand into a template strand. Once we have the template strand, we are then able to transcribe the strand into an mRNA strand. With the mRNA strand, we're now able to translate the strand into our peptide chain. With no mutations, this sequence translates into this. This stop codon indicates that the peptide chain translating has finished, and this chain can be used for various functions throughout the body. If we now look at point mutations, we see that there are four types: missense, nonsense, silent, and read-through. This first type is a missense mutation, which is a single mutation in one of the DNA pairs that changes the corresponding amino acid, which can have varying degrees of effectiveness, which can be as extreme as making a protein non-functional. For example, in the coding sequence, we can change this nucleotide from a cytosine to a guanine. This will change the mRNA sequence to change from a UGC to a UGG, which changes the coded amino acid from cysteine to tryptophan. This cytosine to guanine nucleotide change is known as a transversion. The second kind of mutation is nonsense, which is when the codon turns into a stop codon, which shortens the amino acid chain. For example, when changing the DNA sequence here, placing an adenine instead of the guanine, we can see that the original tryptophan amino acid actually now codes for UGA, one of the three stop codons. The changing of the guanine to adenine is a purine-to-purine switch, also called a transition. This is the result because of this mutation. The third style mutation is called a silent mutation, which is a mutation that changes a single nucleotide but actually does not change the corresponding amino acid. If we were to change this nucleotide from a cytosine to a thymine, we can see that the codon changes from AAC to AAT, which are two codons that code for asparagine. This is another example of a transition. The last kind of mutation is called a read-through mutation. This type turns the stop codon into an amino acid, allowing for mRNA strand to continue to be read and elongates the amino acid chain. If we perform another transition, we change this adenine to a guanine and transform the stop codon into a tryptophan, which then allows the next two codons on the mRNA strand to code for proline and glycine.